The main objective of this practice is to understand the correct usage of the equipment in genetics laboratory, and today we're going to focus on micropipettes.
The micropipettes are a special piece of equipment that allow the precise measurement and transfer of liquid volumes in microliters. It is important to understand the proper use of this equipment to achieve optimal results during experimentation. There are several types of micropipettes that depend on the measurement range required. The range of the micropipettes can vary from 2 microliters up to 1 milliliter. The micropipettes can vary in the amount of tips they can hold, mono or poly-type pipettes. These last ones can be used to speed up the testing process. The tip used in the equipment will vary depending on the size and range that the specific pipette is calibrated to.
The correct usage of micropipettes should follow the next steps.
Step 1. Release the lock in the top of the pipette. 2. Adjust to the required volume by rotating the knob. 3. Press the shell tip holder into the tip, applying pressure until it locks in. 4. Press the superior button until the first click and insert into the sample in a vertical motion. 5. Slowly release the superior button until the tip is filled with the sample. 6. In a vertical motion, press the superior button until the first click where the sample will be placed. 7. Continue pressing until the second click in order to purge the pipette. Steps 6 and 7 can be performed in 3 different ways: vertical, slanted, touching the wall, or not touching the wall near the solution. 8. Release the tip using the ejector button and place it on the disposal container.
Activity 1. Through this experimentation, several steps were followed in order to learn the correct handling of micropipettes and how to calculate the error in each one of them.
Step 1. Distillated water was used for 10 different measurements in 3 different volumes on each of the available micropipettes. Step 2. Every measurement was weighted in an analytical balance in order to calculate the error through density. For a 20 microliter pipette, they were measured 2 microliters, 10 microliters, and 20 microliters. For that 200 microliter pipette, they were measured 20, 100, and 200 microliters. And finally, for the 1000 microliter pipette, 100 microliters, 500 microliters, and 1000 microliters were measured.
This activity was based on the recollection of a field volume obtained from minor measurements.
For the P20, 4 measurements of 3.5 microliters were deposited in a microtube, then the total volume was recollected, calculating the error in the previous measurement. For the P200, 3 measurements of 48 microliters were deposited in a microtube, then the total volume was recollected, calculating the error in the previous measurement. For the P1000, 3 measurements of 330 microliters were deposited in a microtube, then the total volume was recollected, calculating the error in the previous measurement. Based on the minor measurements and the recollected volumes, the standard deviation obtained were 0.374, 0.726, and 1.374 correspondingly. In practice, we learned that a correct management of the micropipette is essential to obtain accurate results during experimentation.
We also learned that even if the micropipettes are used properly, there will be certain error that should be taken into account when analyzing the results.
In order to reduce this error, the pipettes should be calibrated and equipment, such as tips, should be stored correctly to avoid the damage.
These activities are essential so that future experimentations are done adequately.